Very good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, in our previous session, we have clearly dealt with the tissue systems. What are the three tissue systems that we have done, ma? Epidermal tissue system, ground tissue system or fundamental tissue system, and vascular tissue system. After which we have looked into the comparative analysis between dicot monocot root, dicot monocot stem, and now it is the time to learn the last comparative analysis, that is dicot monocot leaf. After which we will exercise secondary growth. Of course, secondary growth is not updated in your recent NEET UG syllabus, but then we will look into the secondary growth whichever is required, and we will have a clear analysis on differentiated, de-differentiated and redifferentiated tissues, because these three concepts, that is differentiation, de-differentiation and redifferentiation, will be asked in connection with plant growth and development. Yes, na beta? So that thing we will revise and exercise, after which we will also solve the anatomy previous year questions. Clear, right, ma? So yes, now you see, first is what? Dicot leaf. In dicot leaf you have epidermis, two epidermal layers, beta. The upper one is called as adaxial epidermis, lower one is abaxial epidermis. Both the epidermis is covered by cuticle. Epidermis is cuticularized, which means it is covered with cuticle. Okay. Adaxial epidermis is also called as ventral epidermis. It is on the ventral surface. Adaxial epidermis is on the ventral surface, and it is called as adaxial ventral surface, which is on the ventral surface. This is upper epidermis. Fine, ma. And here abaxial epidermis, which is on the lower side. This is lower epidermis and present as on the dorsal surface. Okay, this is present on the dorsal surface, and this is lower epidermis. Fine, right? So adaxial, abaxial epidermis is clear. What are the components that are present in the epidermis over here? Epidermis consists of stomata. Okay, it consists of, you can say, stomatal apparatus. Stomatal apparatus includes stomatal pore, okay, guard cells and subsidiary cells. Fine. And here in a dicot leaf, more number of stomata are present on the dorsal surface or lower epidermis, and less number of stomata are present in the adaxial epidermis or the upper epidermis. Okay. This is because the leaf which is exposed towards sunlight, that is upper epidermis, right? Here it is exposed towards sunlight. To reduce or to minimize transpiration, more amount of stomata are present on the lower epidermis. Got it, beta? So more stomata are present where? In the lower epidermis. This is an adaptation to reduce the process of transpiration. Here what all are present in the stomatal apparatus? It has stomatal pore. Of course, guard cells are of what shape? Bean shape, right? Okay, guard cells are bean shape, where at the center you have stomatal pore. Guard cells are bean shape. It has two wall layers, outer thin wall and inner thick wall. Okay, outer thin wall and inner thick wall. Based on the turgid and flaccid conditions, the stomatal opening and closing will be there. Okay, that is because of thin and thick wall layers. And the cells which are closely arranged around the guard cells are called as what cells, ma? Subsidiary cells. They are subsidiary cells. Cells closely arranged towards the near to the guard cells are subsidiary cells. Clear, right? Any doubt over here? No. Next, Under the ground, so under epidermal tissue system, here you have upper epidermis and lower epidermis. Under ground tissue system or fundamental tissue system of leaf, you just have mesophyll cells. Okay, you have what? Ground tissue system here is mesophyll tissue system. Ground tissue system is having mesophyll tissue. Mesophyll tissue. Mesophyll tissue is of two types here. What is that? Palisade parenchyma and spongy parenchyma, or palisade mesophyll and spongy mesophyll. Okay, it has palisade mesophyll and spongy mesophyll. Spongy mesophyll. So here you see, palisade mesophyll cells or palisade mesophyll tissue is arranged vertically below what? Adaxial epidermis. Okay, this is perpendicular to adaxial epidermis. These are parallelly arranged. There are less intercellular spaces in palisade parenchyma. When you observe spongy parenchyma, they are loosely arranged as spongy tissue. Okay, and they have air spaces inside them. Okay, and here vascular tissue system includes what? Vascular tissue system includes vascular bundles, which includes what, Nana? Xylem and phloem. Okay, remember, xylem is towards the upper epidermis, phloem is towards the lower epidermis. What type of vascular bundles are present over here? Vascular bundles are conjoined and closed. Fine, right, ma? Any doubts over here in the dicot leaf anatomy? Dicot leaf anatomy? Yeah, you see here, first epidermis. Under epidermal tissue system you have adaxial epidermis and abaxial epidermis. Adaxial epidermis is also called as upper epidermis, which is on the ventral surface. Abaxial epidermis is on the dorsal surface, it is lower epidermis. What are present in the epidermal tissues? Epidermis is composed of parenchymatous tissue. It is cuticularized. Okay, it has what, Nana? Stomatal apparatus, which has stomatal pore, guard cells and subsidiary cells. Guard cells in the dicot leaf will be bean shaped. Okay, it has outer thin wall, inner thick wall. Close proximity of this guard cells will have what? Subsidiary cells. So next is what? Ground tissue system or fundamental tissue system in a dicot leaf is composed of mesophyll tissue. Okay, that mesophyll tissue includes palisade mesophyll tissue and spongy mesophyll tissue. Palisade mesophyll tissue is vertically arranged parallel to each other with least intercellular spaces. Okay, next is spongy mesophyll tissue. Spongy mesophyll tissue, where mesophyll tissue is loosely arranged with air cavities inside them. Okay, and vascular bundles are also arranged inside the spongy mesophyll tissue. Vascular bundles consist of xylem and phloem. Xylem is towards the palisade parenchyma, phloem is towards the lower side. Okay, that is lower epidermis. Fine. And what type of vascular bundles are there, ma? Conjoint and closed type of vascular bundles. Okay, so in a dicot leaf, now you tell me, in a dicot leaf, just roughly I will write, first is what? Epidermal tissue system. Consists of what, ma? Adaxial and abaxial epidermis. Okay, here in the abaxial you have more number of stomata and both of them are cuticularized. Fine, right? Next, second is ground tissue system or fundamental tissue system will have what? Mesophyll tissue. This mesophyll tissue is two layered, arranged in two types of mesophyll tissues are there. First is palisade parenchyma, which is arranged in 2 to 3 rows below the upper epidermis. There are least intercellular spaces beside it. And second is spongy parenchyma. Okay, spongy parenchyma will have air spaces inside them and vascular bundles are placed inside the spongy parenchymatous tissue. Next is what? Third is vascular tissue system. Includes xylem and phloem. How they are arranged? Conjoint and closed. Simple, right? Simple, right, ma? Any doubt anywhere? No, right? Yeah. Next. Monocot leaf. It is much simpler than the dicot leaf's anatomy. Monocot leaf also has adaxial, abaxial epidermis. Okay. Adaxial epidermis is present. Abaxial epidermis is present. Both of them are cuticularized. Okay. Both are surrounded by or covered by cuticle. Both are covered by cuticle, adaxial and abaxial epidermis, and equal distribution of stomata. Unlike dicot leaf, where more stomatal distribution is on the lower surface of the epidermis, here you have equal distribution of stomata. Equal distribution of stomata. Fine. Next. Second is ground tissue system consists of just one type of mesophyll tissue. Only mesophyll tissue is present in the ground tissue system. Okay. Inside this single type of mesophyll tissue, okay, you have what? Vascular bundles. Vascular bundles include again xylem and phloem. They are arranged in conjoint and closed type. One more type, ma. One more thing I have to tell you here. Peculiar feature or component which is present in the epidermal tissue system. Those are large and colorless. Large and colorless bulliform cells are present. Bulliform cells. Okay. These are the components which you see only in the epidermis of monocot leaf. Okay. Large and colorless bulliform cells. These bulliform cells, what does they do in the epidermis? Whenever the cell or the leaf is in flaccid conditions, in water loss conditions, at that time leaf will be flaccid, right? Cells will get flaccid, right? Then leaves roll towards inner side. Okay. Bulliform cells which are present in the epidermis will facilitate that movement of leaves rolling towards inner side in the flaccid conditions. Whenever the leaf has ample of water, okay, in turgid conditions, the leaf straightens, indicating sufficient water. That is because of bulliform cells. Bulliform cells are present where, ma? In epidermis of monocot leaf. What are the key activities of bulliform cells? First is thank you. During turgid conditions, when cells are turgid, leaf straightens because of bulliform cells. When leaves are flaccid, when cell is in flaccid condition, in water loss condition, at that time leaves leaf rolls towards inner side. Clear? Any doubt anywhere? Simple, right? Simple anatomy when compared to dicot leaf. Once it is done, let me know, ma. We will compare between dicot leaf and monocot leaf of 3 tissue systems. Clear? So here let us compare between dicot leaf and monocot leaf. Have a rough table as it is. First is epidermal tissue system, where here you have cuticle surrounded by cuticle. Here also cuticle is present, yes or no? Here it is 2 layered, adaxial, abaxial. Here also the same case, adaxial and abaxial. Which is present on the ventral surface, ma? Very good, adaxial surface, good. Here maximum number of stomatal distribution is on the abaxial surface. Fine. Here equal stomatal distribution. Fine, right, ma? And here there is one more additional component that is present. What is that? Bulliform cells on the adaxial surface. Okay, on the adaxial surface, important, this is bulliform cells are present. Large and colorless cells. Fine. And then second is ground tissue system. Fine, ground tissue system. Ground tissue system, here if you see, it has mesophyll tissue. This mesophyll tissue is of 2 types, palisade and spongy. Here only one type of mesophyll tissue is present. Here palisade parenchyma is arranged in 1 or 2 rows below the upper epidermis. Spongy parenchyma is arranged towards lower epidermis, which has air spaces in between them. Yeah, and vascular bundles are placed inside that spongy parenchyma. Next. Vascular tissue system. Okay, includes what? Xylem and phloem. Okay, which are conjoined and closed. Fine. Here also the same case. Xylem and phloem, they are conjoined and closed. Here one more added on feature, ma. Surrounding xylem and phloem, okay, more than 2 to 3 layers of bundle sheath cells are present. Bundle sheath cells. where you will learn it under Cran's anatomy, okay, where C4 pathway will be operated, plant physiology, okay. So, bundle sheath cells are present surrounding the xylem and phloem in monocot leaf, okay. These bundle sheath cells have agranule chloroplast, fine, they are operated in C4 pathway. This we will discuss more detail, we have discussed already when we are done with plant physiology, okay. Take down the differences and then we will move on to the secondary growth. Done? Now, secondary growth. Keep the heading of secondary growth. How can you define secondary growth, Asma? Secondary growth can be defined as the growth which happens in the later part of plant life, where increase in the girth of shoot and root is observed, okay. This happens in the later part of plant life. It is because of lateral meristems, okay. It is because of what? Lateral meristems, lateral or secondary meristems will increase the girth of the plant, girth of root and shoot. Fine, right? Now you see, this is about lateral or secondary meristems, fine. What are the lateral or secondary meristems? As such, you can see two types of growth. Firstly, you observe what are those meristems. First is cork cambium and second one is vascular cambium. Vascular cambium, if it is present in between xylem and phloem, you address it as what? Intrafascicular vascular cambium or fascicular vascular cambium, okay. And if it is present in between two vascular bundles, it is said as interfascicular vascular cambium. Fine, interfascicular vascular cambium. Cork cambium will conduct extrastellar secondary growth. Extrastellar secondary growth. Whereas vascular cambium will conduct stellar secondary growth because it occurs in the stele region. Fine, right? Take down this. I will give you a brief anatomy on this, like how secondary growth is happening. Done? Here you see, ma. Up till here, if you know, it is enough, ma. Okay, sapwood, hardwood, autumnwood, springwood, not required. Okay, not updated in your syllabus yet. So remember one thing, a small gist of this. So outer layer you will have epidermis. This we know, no? Epidermis. Below epidermis we have what, ma? Hypodermis, general cortex, endodermis. Yes or no? So the next region is completely cortex region. This completely is what? Cortex region. Last layer of cortex is what? Endodermis. And then comes the Stellar region. Stellar region first includes what? Pericycle. Stellar region first includes pericycle. Fine. So the outer layer is pericycle. If you consider the stem, stem has what type of vascular bundles? Conjoint and closed. Conjoint and closed vascular bundles, where xylem will be towards the pith, phloem will be towards the periphery. Okay, and this is intravesicular cambium, this will be intervesicular cambium. This forms a cambial ring, yes or no? Intravesicular cambium and intervesicular cambium. This conducts what activity? Meristematic activity. Those cells which are formed towards the xylem are called as what? Secondary xylem. Okay. Those cells which are formed towards the phloem are what? Secondary phloem. Fine. So if you consider the stellar region, at the center you have pith, then primary xylem, followed by secondary xylem, then vascular cambium, followed by secondary phloem, and then primary phloem. Primary phloem is close to pericycle, yes or no? Yeah. Now you see the extrastellar secondary growth. In cortex region also, few of the cells will become meristematic and they form what? Cork cambium. Okay, few of the cells are getting meristematic, they form what? Cork cambium. Cork cambium also called as phellogen. Fine. Those cells, new cells which are formed towards the epidermis are called as what? Cork cells. Cork cells are also called as, or cork is also called as phellum. Okay. Cells which are formed towards the endodermis is secondary cortex or phelloderm. Phellum, phellogen and phelloderm together called as what? Periderm. So clear beta. So here if you have to see from epidermis to pith, if you have to analyze the secondary growth, first epidermis followed by cork cells or phellum, cork cambium, secondary cortex, endodermis, pericycle, primary phloem, secondary phloem, vascular cambium, secondary xylem, primary xylem and pith. Just to take that flowchart, it is enough. The gist of secondary growth will be in your mind. Okay? Yeah, take down. It is from epidermis followed by cork or phellum, followed by cork cambium, then secondary cortex, followed by endodermis. Fine. Endodermis followed by pericycle, then primary phloem, secondary phloem, vascular cambium, followed by secondary xylem, primary xylem, pith. Fine, right ma'am. Here you can see cork cambium, here vascular cambium. Clear, right? Clear is the flow of secondary growth. Any doubts you are having over here? Any doubts, let me know. Done? Now we will see differentiation, dedifferentiation and redifferentiation. Keep the heading. Differentiation, dedifferentiation, and redifferentiation. First you see meristematic tissues. Okay. Meristematic tissues. We will start from here. We have two types of meristematic tissues based on position. What are they? Apical and intercalary meristems. Apical, again we have two types, shoot apical meristems and root apical meristems. Okay. Here you see these meristems will divide and divide. What type of division? Arithmetic division, where one cell divides into two, of which one continues the division and the other gets permanent. Okay. Again this continues the division, other gets permanent. Okay. Second is what is? Okay. Second is, first is meristematic tissues. These cells which are getting differentiated will form differentiated tissues. Differentiated tissues. We have two types of differentiated tissues. What are they? Simple permanent tissues and compound permanent tissues or complex permanent tissues. Simple permanent tissues include parenchyma, collenchyma and sclerenchyma. Complex permanent tissues include xylem and phloem. Now it is our responsibility to find out the regions, differentiated regions. Okay. Regions include epidermis is differentiated, yes or no? Cortex, yes. Endodermis, pericycle, pith, vascular bundles, that is primary xylem and phloem. If you also want to add on, you can also add on primary medullary rays as differentiated tissues. Okay. Primary medullary rays. Clear? These are the differentiated tissues and differentiated regions in the plant. Fine ma. Now you see, of these regions in cortex region and vascular bundles region, few of the cells are getting meristematic, means they are losing the activity of differentiation, they are gaining the meristematic activity. So that is why we call them as what? De-differentiation. So next, third is what? De-differentiation. Includes what regions, ma? De-differentiation includes vascular cambium, and cork cambium. With the activity of their meristematic activity, again few cells are getting differentiated, right? Because the pattern of growth is arithmetic, where one cell divides into two, one will be meristematic, the other is permanent. Again this divides into two, which gets permanent. As such, these cells which are permanent cells which are formed as a result of secondary meristem's activity are redifferentiated tissues. Okay. So fourth is last is what? Last component is redifferentiation. Okay. Differentiation. Ted tissues or regions. Yes, Abdul, any doubt, ma? Tell me. Exactly, very good. Secondary xylem, secondary phloem, secondary cortex, cork tissue, okay, secondary medullary rays, all these comes under what? Redifferentiated tissues or regions in the plant. Very good. So you have cork or phellum, secondary cortex, secondary xylem, phloem. Fine. Look at everything. I will give you 2 minutes of time. If you are having any doubt, ask me, ma. Clear? Clear with the entire chapter? Shall we go on to the objective practice? Online students, you can take a screenshot of this, ma. Complete concept on differentiation, de-differentiation, redifferentiation. The question will not escape from this slide. Okay? Fix this in your mind. Fine? Yeah. We will get into the objective practice. Yeah. Recent asked question NEET 25. Try it out. No problem. I will help you. Huh? Try it out. Online students, even you can answer, ma. Very good evening. Start answering the objective beta. NEET previous year questions. First question. Find the statement that is not correct with regard to the structure of monocot stem. As I told you already, definitely you will get a question on the comparative analysis. See here, monocot stem. Which statement is not in relation with monocot stem? Vascular bundles are conjoined and closed, correct only, no re? Phloem parenchyma is present. Hypodermis, phloem parenchyma is absent, sorry. Hypodermis is parenchymatous, he is saying no. Hypodermis in monocot stem is what? Sclerenchymatous. Good. Hypodermis in monocot stem is sclerenchymatous. Cholenchymatous in dicot stem. Vascular bundles are scattered in the cortex region. So what is the wrong statement here? Yeah. Next question, ma. NEET 2024. Bulliform cells are responsible for, just now we have discussed. Just now. Inward curling of leaves, no? There is no role of photosynthesis for bulliform cells. Protecting the plant, no, there is no role in salt stress. Inward curling of leaves is done when there is water stress. Okay, when there is water loss conditions, flaccid conditions, leaves curl towards inner side. Providing large spaces for storage of sugars, no, it has no role for that. What is the right answer, ma? Next. NEET 2024 again, statement based. Statement 1 you see, parenchyma is living but chorenchyma is dead tissue. No, both are living only. Sclerenchyma is only dead tissue. Wrong. Gymnosperm lacks xylem vessels but presence of xylem vessels is characteristic of angiosperms. True. Vessels are absent in the xylem component of gymnosperms. When phloem is considered, what is absent in gymnosperms? When phloem, in phloem tissue of gymnosperms, what are absent? Sieve tube cells are absent. Rather, in the place of sieve tube cells, only sieve cells are present. Companion cells are absent. In the place of companion cells, albuminous cells are present. Okay, remember this. Vessels are absent from xylem in gymnosperms and it has sieve cells and albuminous cells. Doesn't have sieve tubes and companion cells from phloem. Okay, so here statement 1 is wrong, 2 is correct. 1 is false, 2 is true. 1, okay. And one more thing to clear, secondary growth happens in dicots. Okay, dicots class of angiosperm, secondary growth happens. Even in gymnosperm, secondary growth occurs. Only in monocots of angiosperm, secondary growth is absent. Remember, secondary growth occurs in gymnosperms and dicots of angiosperms. Write down that statement and answer this question. Secondary growth occurs in dicots of angiosperms and gymnosperms. Now you look into this question, this one. Assertion you see beta. Late wood has fewer xylary elements with narrow vessels, correct. Cambium is less active in winters. True. Both A and R are true, but R is nowhere explaining assertion. Don't worry, these questions will not come because secondary growth is not there in your current syllabus. Next question. Statement 1, endarch and exarch are the terms often used for describing the position of secondary xylem in the body. No, these are the terms which are often used to describe primary xylem, position of primary xylem. Okay, in primary xylem, the initially formed xylem is called as protoxylem beta. Matured one is called as what? Metaxylem. If protoxylem is towards periphery, it is exarch. Yes or no? Seen in root or stem? Good. If protoxylem is towards pith, It is called as endark. Fine, observed in? Got it? Yeah. So 1 is wrong. 2. Exarch condition is the most common feature of the root system. 1 is wrong and 2 is correct. Which is the right option, beta? Good. Next. You can take a note of this if you want. Keep the heading NEET PYQ 2023 and write down this as a brief. Will go good for your revision, huh? Done, ma? Here you see again statement based. So if you have to analyze NEET PYQs, most of the questions are of statement based only. Look at this, ma. Identify the correct statements. This is also from secondary growth, not required. This is from, yeah, this you can answer. This is also from secondary growth, asked from bark elements. Yeah, you can answer this. क्यों हंस रहे? हाँ? You got the answer or not? Done? Shall we go? Yeah. See, read the following statements about the vascular bundles. In roots, xylem and phloem in vascular bundle are arranged alternate manner at different radii. That is why it is called as what? Radial type of vascular bundles. Next, conjoined closed vascular bundles do not possess cambium. Correct? In open vascular bundles, cambium is present between xylem and phloem. True. The vascular bundles of dicotyledonous stem possess endark protoxylem. Okay. In monocot root, usually there are more than 6 xylem bundles. Yes, monocot has polyarch xylem bundles. All the statements are correct. Yes or no? Any doubts, anyone? This is also on wood. And this question you answer. Cells with active cell division capacity? Yes or no? Meristematic tissue. Tissue having all cells similar in structure and function are simple permanent tissues. Tissues having different types of cells. Vascular tissues, that is why complex permanent tissues, no? Dead cells with highly thickened walls and narrow lumen sclerites. Now you tell me what is the right answer? Yeah. Already told ma'am, huh? This one. Lenticels, exchange of gases. See, because of secondary growth, cork cells are formed, right ma? Epidermal cell will be ruptured. As such, cork cells will move on to the epidermal cell. Small pores are created. Those are called as lenticels. They act as breathing pores of the stem. Fine. So, lenticels for exchange of gases. Okay. Cork cambium, secondary cortex. Just a second. 3. Phylloderm. Cork is suberine depositions. Cork has suberine depositions. So, what is the right answer? That's it. Look at this. Simple, you can answer. Statement based again. 3. What is the correct pair? Cells of medullary rays that form part of cambial ring are interfascicular cambium. True only. In dicot leaves, there is no conjunctive tissue in the leaf. Large cells, large colorless empty cells are what? Bulliform cells, not subsidiary cells. Loose parenchyma cells rupturing the epidermis, forming lens shaped, that is called as lenticels, not bark spongy parenchyma. So, it is option C. Good Abdul, very good. This one. The transverse section of plant shows following anatomical features: large number of scattered vascular bundles, conspicuous parenchymatous ground tissue, vascular bundles conjoined and closed, phloem parenchyma absent. It is very simple to identify. What is it talking about? Agreed or not? See, scattered vascular bundles, large conspicuous parenchymatous ground tissue, vascular bundles conjoined and closed, phloem parenchyma absent. This you have already answered. That's it. That's it with NEET previous year questions. So, we have done with the anatomy of flowering plants. Once you check it out, we have completed plant physiology, yes or no? Photosynthesis, respiration, plant growth and development. We have completed cell biology, cell the unit of life, cell cycle, cell division and biomolecules. Then we have done with living world, biological classification, plant kingdom, morphology, anatomy. With this, from botany end of 11th NCERT syllabus is done. Okay. And then we will move on to 12th NCERT, where I will be starting sexual reproduction of flowering plants from our next class. Okay, Navita? Yeah, that's all for today's session. Thank you.